Okay, so we're going to do a session on the carbohydrate chemistry. We're going to take a look at monosaccharides, disaccharides, polysaccharides, and the formation of disaccharides and the various monomers that are involved in the formation of polysaccharides. So, basic recap of the first section of Unit 1 for Excel Biology B. So, word I need you all to be familiar with is the word saccharide. It means sugar. So when we hear the word saccharide, it means sugar. Saccharin also means sweet. So it's what the root of the word means. So when you hear saccharide, think sugar. Monosaccharides. Now, you need to think back to the chemistry that you've done, organic chemistry, when you think about monomers and polymers. Monosaccharides are the monomers for the production of polysaccharides. The monosaccharides that you need to know about are glucose. So that's a simple sugar. The other one is fructose. Got lactose, ribose, and deoxyribose. Okay, these are all monosaccharides that are referred to in the course, and you need to have an idea of what each of them are involved in. When we think disaccharide, di means two, so two sugars, so it's this two sugars joined together to form a disaccharide. The disaccharides you have to have an awareness of are maltose. We'll talk about the formation of maltose a little later. Sucrose and lactose. When we think of polysaccharides, poly means many, so many sugars joined together. The one that everybody will be familiar with is starch. Now, starch is interesting because actually, when we think about starch, starch is mainly composed of two types. The two types are amylose and amylopectin. Think about, from GCSE, when you think about starch digestion, it's carried out by the enzyme amylase. When we see AMY, that basically tells us that it's something to do with starch. So when we think about in the cell, in plant cells, amyloplasts are the organelles involved in the formation of starch. The other polysaccharides we will be looking at are cellulose and glycogen. Glycogen is similar to starch. It is composed of lots of alpha glucose molecules. Cellulose is an interesting polysaccharide in that it's made of beta glucose molecules. We will look at the difference between these three polysaccharides in a bit more detail. So the next thing I want you to be aware of is then the fact that glucose comes in these isomers. There are two isomers of glucose you need to know. That's beta glucose and alpha glucose. So we'll start with alpha glucose. It's important. We'll come back to its importance a little bit later. But you must be able to draw the structure of alpha glucose and see where the functional units are that allow it to do what it does in the formation of maltose and so forth. So my little strategy for drawing it out. So give yourself plenty of room and make sure that you spend plenty of time practicing this. I'll start with carbon 5. So there's carbon 5. And it has a bond to an oxygen molecule. And then I do carbon 6, and it has 2 hydrogens and an OH group bonded to it. So we show it in that way. Coming off from carbon 5, there is a bond to carbon 4. From carbon 4, there's a bond to carbon 3. From carbon 3, there's a bond to carbon 2. And from carbon 2 to carbon 1, and the oxygen bonds to carbon 1. We then need to fill in the rest of the molecule. So at carbon 5, there's a bond with a hydrogen. At carbon 3, there's a bond to an OH group here, and a hydrogen here. For carbon 2, there is a bond to a hydrogen here, and an OH here. Carbon 1, there is a hydrogen here, and an OH group here. And carbon 4, OH group here, hydrogen here. That is alpha glucose. The important bits of alpha glucose that you must get in the correct positions are this OH group at carbon 1 and this OH group at carbon 4. We might also spend a bit of time talking about this OH group later on when we talk about glycogen and amylopectin. Now it's important at this stage you know the carbons and how they're numbered. So this is carbon 1, 2, 3, 4, 5, and 6. We'll come back to that a little bit later. But again, this is alpha glucose. It is the monomer involved in the formation of maltose, a disaccharide. It's involved in the formation of lactose, another disaccharide. It's involved in the formation of sucrose, another disaccharide. Then it's involved in the formation of starch and glycogen. So it's a really important carbohydrate. So moving on to beta glucose. So this is beta glucose, and it's very similar, slight difference, so make sure you are aware of that. So I'm going to draw out the molecule again, starting again with carbon 5, bonded to carbon 6. So it's bonded to the oxygen, and we get that hexagonal kind of shape going on again, from carbon 4 to carbon 3, to carbon 2, to carbon 1. We can put in the little h here, we can put in the OH group here, the hydrogen here, the hydrogen here, the OH group here. Now, this is the bit where you need to pay attention to the difference. So, on carbon 1, we have an OH group like that, and a hydrogen like that. On carbon 4, we have an OH group here, and a hydrogen there. This is beta glucose. The difference that I need you to see here is that we have the OH group here like that, and again down here like that. Having that OH group there means that that there's implications for the formation of a bond between another beta glucose molecule, which we'll talk about when we discuss the formation of cellulose. Now, one of the things that I've negated to say so far, and if I just flip back so you can compare the structure of the two. Side by side there, we can kind of see them together. Both of these are hexose sugars. Just spend a minute on that. Hexose sugars. They're both hexose sugars, which means they both have 6 carbons. So when we think about being a hexose sugar, the molecular formula is C6H12O6. Hexose sugar, as in there are 6 carbons. In a second, we're going to look at ribose, which is a pentose sugar, and it's got 5 carbons. The other thing to note is that if we look at galactose, or if we look at fructose, if we look at alpha or beta glucose, they all have this molecular formula, but they're all isomers in the sense that they all have different structures. So make sure you spend a bit of time looking at the meaning of this word isomer. You will have loads of fun with this whenever you do A-level chemistry as well. So moving swiftly onwards, wants to spend a bit of time taking a look at a couple of other important molecules. Just to bear in mind as well that beta glucose and alpha glucose are absolutely essential to know the structures of. You will be assessed on them potentially. The next one that is important for you to have a grasp of is ribose. This is also mentioned on the specification, and it's another sugar that you must be able to draw the structure of. You will have potentially heard of ribose. I would imagine you will have definitely heard of deoxyribose. These monomers, or these saccharides, monosaccharides, are important firstly for the formation of RNA. Ribonucleic acid, so the sugar in ribonucleic acid is ribose, and in DNA it's deoxyribose. So looking firstly at the structure of ribose, as I said a minute ago, ribose is a pentose sugar, so it is C5H10O5. So we start with an oxygen at the top of the molecule, and it's 2 bonds bonding to carbons. These carbons then bond to other carbons like that. So far we have 4 carbons as part of our ribose. And carbon, this is carbon 1, this is carbon 2, this is carbon 3, carbon 4, carbon 5 is up here. With our H2OH group on it. At carbon 4 here we have a hydrogen. At carbon 3 we have an OH, and at carbon 2 we have an OH, which means we have a hydrogen here and a hydrogen here. And then for carbon 1 we have a hydrogen here and an OH here. So carbon 1, 2, 3, 4, and 5. Carbon 5 and carbon 3 are really important. They allow the formation of bonds to other ribose and phosphate groups to allow the backbone of the mRNA molecule or the tRNA molecule, which we will talk about in a bit more detail in a later lesson. But this is the structure of ribose. Now, you have mentioned deoxyribose is another really important sugar. Both ribose and deoxyribose are involved in the formation of nucleic acids, ribonucleic acids for RNA and deoxyribonucleotides for DNA. They are really similar, and I just need you to have an awareness of what the difference is. So doing deoxyribose underneath, exactly the same structure. So we start with the oxygen with its bonds to carbon 4 and carbon 1, with its bond to carbon 3 and then carbon 2, and then going off to carbon 5. The rest of the molecule so it's all looking exactly the same so far, but here's the crucial difference. At carbon 2, instead of an OH group, we have a single hydrogen. So it's missing an oxygen, and that's what deoxyribose means, without an oxygen at this position here. So this is deoxyribose. And its formula is C5H10O4. So moving on, I want us to spend a minute or two looking at the structure of a couple of other molecules. Now, you will not be required to draw the structure of fructose, but I think it's worthwhile to spend a minute taking a look at the structure so you can appreciate the molecular formula and how it's different from the structural formula. So fructose is another hexose sugar, so again it has the C6H12O6, and we start with an oxygen with its bonds going off to a couple of carbons. So we have a carbon here, and it's got that sort of pentagonal kind of shape, but we only have 4 carbons so far, and as I've said, it's a hexose sugar, so carbon 1 is sitting down here and carbon 6 is sitting up here. So we have the H2OH on here and the H2OH on here. At carbon 2 we have the important OH group. It's this OH group which is involved in the formation of a bond between another alpha glucose molecule to form sucrose. And if we look at the rest of the molecule, we would have a hydrogen here. We would have hydrogen here, so an OH group here, and an OH group here, then a hydrogen here. So it's worthwhile to spend a minute making sure you understand the structure of that, and then it will obey the molecular formula of C6H12O6. Like I said, this is an important monosaccharide, and it's involved in the formation of sucrose. We'll look at that in a bit later. Now there's one more monosaccharide I want to spend a bit of time with you on, and that is galactose. Galactose is very similar to alpha glucose. Again, the little star there means that it's not one that you have to learn the structure of. It's worthwhile just knowing, because it's involved in the formation of lactose, and the name gives that away slightly. Like I said, very similar to alpha glucose. So I start with carbon 5, going to the oxygen, carbon 6. So we come down onto carbon 4, 3, 2, and 1. So we'd have the little hydrogen here, an OH here, hydrogen here, hydrogen here, OH here. So at carbon 1, where we get the business end of the molecule, we'd have an OH group here. So it's looking a little bit like beta-glucose at the moment. At carbon 1, if we go over to carbon 4, we would have an OH group here as well. This is galactose. This is an animal sugar. It's found in milk. And when lactose is digested, a hydrolysis reaction occurs to break the lactose molecule into alpha glucose and galactose, which we can then metabolize. Now, moving on, the next important thing to know is how disaccharides form. So disaccharides form because of condensation reactions. That's a chemical reaction where a water molecule is produced. We'll look at a specific condensation reaction in a moment. But in the spec, there are 3 disaccharides to know. The first one is maltose. Maltose is a disaccharide formed from the formation of alpha glucose plus alpha glucose. So the word equation, which is important to know, so we have alpha glucose plus alpha glucose forms maltose plus a water molecule. Don't forget to leave that out. The second is sucrose. She's formed from an alpha glucose plus a fructose to form sucrose plus water, because again it is a condensation reaction. Sucrose is a plant sugar. It's what's transported around in the phloem. So it's an really important biological molecule. We will be familiar with it because it's what we sprinkle on food and sometimes referred to as table sugar. The third is lactose, which is a milk sugar. So that's produced in the milk by cells making milk in the mammary glands. And it is composed of an alpha glucose plus a galactose to form lactose plus the water molecule again. Again, a disaccharide, and it's a milk sugar, so we'd find this in animals rather than in plants. Important to remember, all formed by condensation reactions. So we will look then, the last part of this video is going to be then looking at the condensation reaction that occurs between alpha glucose and alpha glucose to produce maltose. So we start by drawing out the alpha glucose molecule again, so I'll do that quickly because you see me doing that. So we have a main strip skeleton appearing there. Okay, so there's one alpha glucose molecule. What we then need to do is draw another one right beside it. So let's start with the OH group bonded to carbon 4. So there's carbon 5 and carbon 6, carbon 1, carbon 3, carbon 2. Okay. Spend a bit of time practicing drawing those two molecules side by side, and it will be important to get your head around that. What I need you to see is that there's an OH group and an OH group lined up, facing off against one another. And this is a condensation reaction, so think production of water. And we're going to draw a little square around the two hydrogens and the oxygen there, because that will indicate the water molecule that gets produced during this condensation reaction when these two alpha glucose molecules are joined together to form one disaccharide. So there's the H2O produced via condensation. The resultant molecule then will be a molecule of maltose. So if I just draw that out again quickly, because the important bit is to see the formation of the bond, the glycosidic bond that's going to form at this position here. So this is the really important bit. So this is carbon 1, and it has formed a bond with carbon 4 here. Okay, now this is the really, really important bit. Carbon 1, 2, 3, and 4 bonded to carbon 1. Just so you can see. So we have a 1,4 glycosidic bond. That's what this bond is called. You may sometimes hear it referred to as an oxygen bridge, but we always would refer to this as a 1,4 glycosidic. Massively important, this. You've got to be able to identify this from drawings. You've got to be able to recall that name, glycosidic. Glycosidic bonds are the bonds that we see in carbohydrates. So starch molecules are held together by glycosidic bonds. Disaccharides are held together by glycosidic bonds. And there's usually the one that we're talking about here is a 1,4. Sometimes we can have a 1,6 and so forth. But let's discuss that in a bit more detail. Now, the very last part of this video is to make sure that you understand what the formula, the molecular formula of a disaccharide is. A little way of making you think about this is that what we've just done is we've added together two maltose molecules. So we think about the structural, or the molecular formula, sorry, of maltose, C6H12O6 plus another C6H12O6, and we were to add them together, we would get C12 H24O12. But don't forget that what we've done is we've extracted or subtracted a water molecule. So if we were to add an H2 and an O and subtract, we would have C12 H22O11. So this is important for when we think about the molecular formula for maltose, lactose, and sucrose. The formula, just write them down: maltose, lactose, and sucrose. We have C12, H22, O11. Okay, so at that moment in time, that's the first stage of our introduction to carbohydrate chemistry. Thank you for watching. Check in again soon, and we will take a look at how these molecules then lead to the formation of polysaccharides. See you soon.